Hello everyone, this is R. Rajarajeshwari working as an assistant professor in the Department of Biotechnology, Kamaraj College of Engineering and Technology. Today we are going to see about nanoparticles that could be used as non-viral transfection agents. So basically there are many types of nanomaterials that could be used for drug delivery like dendrimers, polymeric nanostructures, liposomes, graphenes, metal nanostructures and nanotubes, where some of the targeting ligands like aptamers, monoclonal antibodies, peptides, hyaluronic acid, folic acid and carbohydrates could be integrated into these nanomaterials, and the drug of interest or the genetic material of interest will also be integrated, facilitating intracellular delivery. So there are some of the materials that could be used as non-viral transfection agents. So let's break down the term non-viral transfection agent. What is transfection? First, the introduction of genetic material inside a cell is called transfection. So since we are using these kind of materials for this process and bypassing the use of viral vectors, we are going to call this as non-viral transfection agents or non-viral transfection mechanism. So the materials that could be used for this process include liposomes, chitisins and polymers, and some of the block copolymers abbreviated as DOSPA, DOTMA, DOTAP and DOX. These are the materials that could be used for this process. So to understand more about this non-viral transfection agent, there is a case study which is framed where the objective is to develop a chitisin based nano carrier for delivering a plasmid DNA that could encode for insulin, which could be introduced into the pancreatic cells of mice diabetic mice as a non-viral gene therapy approach. So the methodology for performing this process includes, firstly we have to choose the kind of material that we are going to use and the type of plasmid DNA that we are going to use. Here the material we are going to use is chitisin and the plasmid DNA that we are going to choose is PDX1. So first step in this methodology is the chitisin nanoparticle formation, where the low molecular chitisin is dissolved into the acetic acid and the plasmid DNA PDX1 is introduced into it. So this DNA is going to interact with the chitisin via electrostatic attraction, which will bind well with the chitisin. So since the nanoparticle is formed now along with the plasmid DNA, so next step is to deliver this nanoparticle inside the cell. There is a mechanism called proton sponge mechanism. So let's get deep into it. So before the chitisin enters into the cell, the plasmid DNA is protected well by the chitisin. Since the chitisin is cationic and the DNA is negatively charged, they will bind together well, and this chitisin is going to protect the nanomaterial very well inside the body. So this material has to enter the cell now. So once it enters the cell, proton sponge mechanism is followed. So for entering into the cell, the first step is endocytosis, where the material is taken up by the cell and it is delivered inside the cytoplasm. So after that, this material is going to enter into the endosome. So inside the endosome, there is completely acidic environment where many number of protons are present. So basically this chitisin has amine group, that is NH2 group, that is going to take up all these protons, and hence more water is introduced into the endosome. So introduction of more amount of water creates an osmotic pressure. So this osmotic pressure is created due to the more amount of water reflex inside the endosome. So this step is very critical because this water reflex is going to break the endosomal membrane. Along with that, the chitisin is also going to get broken down, and the plasmid DNA that is integrated into the chitisin is going to get released into the cytoplasm. This is very important step because the plasmid DNA must not be degraded anywhere here. So once this step is completed, the plasmid DNA that is delivered into the cytoplasm is taken up to the nucleus, where further process can be done. So there is next objective where we are going to compare two block copolymers called DOTMA and DOX. for the same case, for delivering insulin related gene into a diabetic mice in the pancreatic cell, and we are going to compare which polymer is going to work best on this. So the polymer chosen are DOTMA and DOX. So these DOTMA and DOX are cationic lipids, and they are going to electrostatically bind to the plasmid DNA molecule that we are going to introduce. So basically every steps will be the same. Let's get into the steps. So first step is liposome preparation, where DOTMA based liposomes and DOX based liposome both are going to prepared by thin film hydration method followed by extrusion process, which ensures uniformity of uniformity in the size of the liposome that are going to be formed. So next step is pDNA and lipid complex formation. This is the step where the required pDNA, that is the plasmid DNA, is going to get integrated into the block copolymer that we are going to use. So next step is cellular internalization. So due to the cationic charge, both DOTMA and DOX are going to interact with the negatively charged phospholipid bilayer of the cell and cell membrane, and they are going to be taken up by endocytosis process. So for DOTMA, the endocytosis process that is going to be followed is clathrin mediated endocytosis, and for the DOX, the method that is going to be followed is macropinocytosis or caveolus mediated endocytosis. So once this is once both the molecules are taken up into the cell, they are going to enter into the endosome where acidic environment is present. So the next target is to escape the endosome, and the plasmid DNA has to be delivered to the nucleus. So for DOTMA, if we see, it is going to follow proton sponge mechanism, because DOTMA has amine groups that will take up all the protons that are going to be present in the endosome, which will introduce more amount of water into the endosome. So due to the more amount of water reflex, osmotic pressure is going to be built up, and the endosome will break along with the destruction of the structure of DOTMA, and the plasmid DNA is getting released into the cytoplasm. But compared to DOX, DOTMA is going to have less efficiency in delivering the plasmid DNA to the nucleus. So if we are going to see in DOX does not need any proton sponge mechanism, because as the name suggests, DOGS, S stands for spermine. The spermine group is going to directly integrate into the endosomal membrane, and it will break the endosomal membrane. So it facilitates easy delivery of plasmid DNA into the cytoplasm. So if we comparatively see about both DOTMA and DOX, DOX is going to deliver the plasmid DNA in a more efficient manner. Here the next step is, once the plasmid DNA is released into the cytoplasm, it has to be delivered into the nucleus. So the release mechanism is followed, and if we see, due to the presence of spermine groups, this DOX the plasmid DNA that is carried to the cell through DOX is going to get into the nucleus through the nuclear receptors, and they are going to interact with the genetic material that are present inside the nucleus, and protein expression is seen. So comparatively, DOX is going to have higher performance. So to summarize the process that we have seen, so to use the non-viral transfection agent for delivering a genetic material, first we have to fix the target. That is, we have to fix the target disease that we are going to treat, and the target body part in which the genetic material has to be delivered. And the next step is choose your genetic material. So we have to decide on what genetic material we are going to deliver. And we have to encapsulate the plasmid DNA in any of the non-viral transfection agent that we have seen, and we are going to introduce a new step called PEGylation. PEG is polyethylene glycol, which will reduce the immunogenicity, that is, it facilitates the escape of the nanomaterials from the immune system, and it will help the molecule reach its target site. And after that, targeting ligand is attached, and once it is done, we are going to decide the route of administration. Thank you for listening.